Hi guys, I just want to start off by saying I'm really sad that I'm not going to get to see you guys in class anymore this year, but we still have to learn things. So some of you guys have been sending me questions about Punnett squares and things you don't understand, and I know I gave you guys the cheat sheet and pretty much everything I'm going to say today is from that, but I know some of you do better when you hear it and see it. So I have my little makeshift whiteboard here. It's a lot smaller than the one we have in class, but we're gonna make it work. So we're starting genetics, and if I asked you guys what genetics was, you'd probably say it's what makes you up, stuff like that. So we're gonna be talking about genes, and genes are really just traits, and all traits have different forms, and they're called alleles. Now, all of the genetics that we're going to be talking about for today are what we call Mendelian genetics, which means they follow the basic rules that Mendel proposed when he was first looking into genetics. And that means that all of the alleles that we're going to look at have only two forms. Now, most things have more than two different alleles. Your eye color, your hair color, your height, all of those things are what we call polygenic. They have multiple alleles that come into play, and we'll talk more about that, not this upcoming week, but the next. So for now, what you need to know are these are the simple things. Only two alleles, okay? So when there are only two alleles, one of those alleles is going to be dominant, and it will be represented by the capital letter. And one of those alleles will be recessive, and it will be the lowercase letter. Now, it's important to know that dominant wins always. If you have a capital letter in a set of alleles, it is always going to show the dominant trait. The only way you can have a recessive trait shown is if you have two lowercase letters. So, a couple more terms we need to talk about. Homozygous and heterozygous. Let's go ahead and give ourselves a letter to talk about. Let's pick a trait from the traits thing you guys did the other day. Let's do tongue rolling, okay? So if you can roll your tongue, you're dominant, okay? So I'm gonna talk about tongue rolling. So I'm gonna use the letter T, okay? You're always gonna wanna use a letter that corresponds to the dominant trait. So for tongue rolling, which is our example that we're gonna use, I can't spell tongue today. For tongue rolling, we're gonna use capital T for dominant, lowercase t for recessive. So in this situation, you can have homozygous, which means same alleles. So you could have homozygous dominant, which would be, in this case, two capital T's, or you can have homozygous recessive, which would be big T, little t. And then your heterozygous form, there's always only one heterozygous. Heterozygous it means different alleles. And in this case, you would have big T, little t. Those are your first terms, okay? I'm gonna erase this because clearly I'm out of room. The other things that we're gonna talk about are the genotype and phenotype. So, the genotype is your allele combinations or letter combinations. So for tongue rolling, like we just talked about, you could be homozygous dominant, which is capital T, capital T. You could be homozygous recessive, which is little t, or you could be heterozygous, which is big T, little t. Then you have the phenotype. The phenotype is the appearance, what it's going to look like in the cross. So for the appearance, big T, and big T, little t, since they have the dominant trait, would both be tongue rolling. And then your other option, recessive, little t, those would be not tongue rolling. All right, so hopefully those basics make sense enough now that we can do an example. I'm going to do a tongue rolling example real quick, and then I'm going to do number 1 on the worksheet that you guys have from last week that's due Monday. I know some of you have already done it and turned it in, and that's great. This really is for the people who were still struggling. Okay, so if you've done it, you've turned it in, you understand everything I've said, you could probably tune out now. But if you're still struggling, I'm going to go ahead and do 2 examples. So let's do a tongue rolling example first. Let's say that parent 1 is heterozygous for their tongue rolling. What that means is their letters are different. So they are big T, little t. And let's say that we want to see what would happen if they mate with someone who is a homozygous non-tongue rolling. So if they're homozygous, that means their letters are going to be the same. If they can't roll their tongue, that means it's recessive. So they're going to be lowercase t, lowercase t. So when we do the Punnett square, you're going to make your box. You're going to put parent 1 across the top, big T, little t, and parent 2 across the side, little t, little t. Now, just a formatting thing when you fill out the boxes, you always want to put the capital letter first if it's there. So the first box, we cross, you have big T, little t. The second box, we cross little t, little t. The third box, big T, little t, and the final box, little t, little t. From there, we determine the genotypes and phenotypes. So the genotypes, you have half big T, little t, and half little t, little t. And from that, you figure out the phenotypes. In this case, you have half tongue rolling and half non-tongue rolling. Now, one thing that is really important to understand here that kind of gets lost when we're not talking about it in the classroom, so I want to make sure I point it out. This is probability. So this does not mean that if these 2 people had 4 children, 2 of them would roll their tongues and 2 of them would not. What this means is that each individual offspring has a 50% chance of being able to roll their tongue and a 50% chance of not. These 2 people could have 10 children and all of them could roll their tongues. They could have 10 children and none of them could roll their tongues. This is just the chance that each individual child would be able to do that. All right, so go ahead and get your worksheet out or pull it up or whatever to do question number 1 together. So, question number 1, if I'm looking at the worksheet, says a green pea plant, and it's so nice to tell me that it's big G, little g, so just to make sure we're understanding, that would be heterozygous, is crossed with a yellow pea plant, little g, little g. So we now know that green is dominant and yellow is recessive. So we're gonna do the cross. Big G, crossed with little g. So you have big G, little G, little G, little G's, little G's. And this one just so happens to turn out the same way that we did before. The genotypes that you have possible are 2 big G, and 2 little g. And the phenotypes that you have possible are 2 green, 2 yellow. So that was an easy one. Sorry, my son knocked it over. Give me a second to make sure we're good. He's not gonna touch it anymore, right? Don't touch it again. Thank you. No. Okay, so I am gonna go ahead and do number 7 with you guys, because number 7, you have to come up with the letters yourself, so I know that's a little more difficult. So in number 7, it tells you a homozygous dominant brown mouse is crossed with a heterozygous brown mouse. Tan is the recessive color. So it's homozygous dominant, so that means it's going to be 2 capital letters. It tells you that dominant is brown, so I'm going to use B's. Okay, so the first parent is capital B, and it's going to be crossed with heterozygous, which means different, so it's got to be big B, little b. Then I do my cross, big B with big B, little b. And then my genotypes that I can have, I've got 2 big B, and 2 big B, little b. Now my phenotypes, notice all of these have a capital letter in the box, which means they are all going to be dominant, which means they will all be brown. These 2 parents could not have a tan offspring. All right, I'm signing off. If you need any further help, let me know. Again, I miss you guys and hope this helps. Bye.